My name is Shriti Pokhrel, and I am a senior at Camas High School in the MST Magnet program. This past summer, I remotely interned with the British conservation program named Operation Wallacea to study and evaluate factors that affect sclerotinia, also known as hard coral, off the coast of Utila, Honduras. For my specific project, I focused on assessing coral diversity at 3 different sites: Coral View, Little Bight, and Sturch Bank, all located off the coast of Util, Honduras. The ultimate goal was to see which sites had the most hard coral diversity and to determine what factors affected these results. So to collect the data, researchers from Operation Wallacea swam alongside a transect at 5 and 15 meters while filming the reef and surrounding areas. This method allowed the scientists to look back at the reefs and to collect data every 25 centimeters. Each depth at every location had 3 to 6 trials, which verified the data to make sure the results were accurate. This is Figure 1, which is a map of the data collection sites. All 3 of the sites are located on the southeast edge of Honduras, with Little Bight being the farthest south and Sturch Bank being the farthest east. This just serves as a visual to show you exactly where data was collected. Moving on, for my part of the research, I used this data and inputted it into R programming software. This is some sample code I used to create the map seen in Figure 1, and later on I used R again to create Figure 2. I used these charts and graphs to compare and contrast the biodiversity at Coral View, Little Bight, and Sturch Bank. I then ran a variety of statistical tests to get numerical data to determine if the difference in location actually played a significant role in the hard coral diversity. The results of the study showed that there was no significant variation in hard coral diversity between the 3 sites. After conducting an analysis of variance test, a p-value of greater than 0.05 was discovered. I observed a p-value of 0.3. As seen in Figure 2, the difference in coral diversity between sites showed no actual significance. Additionally, the standard deviation error bars are almost completely overlapped, meaning that the difference in mean coral diversity between sites does not vary enough to support my initial hypothesis. The outcome of the experiment was different than what was expected. Since there was no statistical significance in the data when comparing location to coral diversity, we cannot attribute any differences in data to outside factors. One probable reason that there wasn't a large difference in the diversity of the coral reefs between the 3 sites is because they are all located fairly close to one another, as seen in Figure 1 from earlier. The factors that affect one of these sites is most likely going to affect the others. This suggests that all the reefs studied are either healthy or damaged, but there is no way of knowing since there are no trends in the data. Another possible reason the results are insignificant is because the overall health of the Mesoamerican reef system has been on a decline since 2006. This means that prior to the decline of the reefs in 2006, the biodiversity in general was greater in the reefs and had a wider variety. Since Operation Wallacea started its research in Honduras in 2012, 6 years after the initial decline, the overall diversity of the reefs decreased throughout the entire region of Util. Of course, this is only a possible explanation, so more research would have to be conducted to prove or disprove this hypothesis. Overall, the results of the study were inconclusive as the data had no statistical significance. Although the data was insignificant, these results can help scientists determine new questions. For future projects, utilizing testing sites that are farther apart may provide better data if trying to compare hard coral diversity and identifying factors that could lead to differences. Since the Mesoamerican reef system stretches over 600 miles, having research sites roughly every 200 miles may yield better results. The issue with this solution is that it would require a lot more funding and more researchers. Narrowing down the research question more and directly comparing different species rather than the mean biodiversity may also be a good option. In all, coral reefs will continue to face the threat of disease and extinction if we don't find a way to protect them. Being able to continue studying these reefs and analyzing their health, as well as biodiversity, is a good next step. I would like to give a big thank you to Mrs. Abraham for helping me get this internship and for guiding me throughout the whole internship process. I would also like to thank my mentor Dan Exton for helping me through the data and all the coding processes. Thanks for listening.